Okay. Now we are going to finally get into our electrical signaling lecture, but I'm starting with the same slide as before because, again, the two, chemical signaling and electrical signaling, are so related and intertwined, and they both have to take place, in these same structures that we've been talking about. So I can start by using the same picture. And I also wanted to start by using the same picture on the chemical signaling in kinases because you guys have a pretty good feel from the lectures that we've been doing about what chemical signaling is, how a cascade of chemical reactions. So because it's in a living thing, these are biochemical reactions where you're activating enzymes and kinases that are just facilitating biochemical reactions, but you can go ahead and, by doing reactions onto the machinery and altering machinery inside cell number 2 here, by altering the machinery and the proteins, because they are molecules. So when you do chemical reactions on those molecules, I actually went through like how that leads to a couple examples of what might be meaningful change to the neuron. So we had a biochemical change lead to the new insertion of new receptors, for example, after a bunch of kinases and enzymes were activated. We went ahead and we inserted these phosphorylated receptors onto the plasma membrane. So boom, there you go. So the other thing I want to say is when we talk about electrical signaling, because you have now a lot under your belt and you have good understanding of chemical signaling, I'm going to try to deliver this lecture by essentially comparison and contrast, because you've done the work and you've got a lot figured out about how this works. That's fantastic, because then I can leverage that to help you understand electrical signaling by comparing and contrasting what happens in electrical signaling. And that's how I'm going to go ahead and do this right now. So the chemical signaling lecture started out by just telling you that we have an electrical signal coming down this axon. This fiber is the axon. And it went ahead and I said that electricity, that electrical signal triggered the release of chemicals, and that kicked off our chemical signaling cascade. But I hinted, I think, at the last question answer video that I gave to you guys, that there had to be coupling between the electrical and chemical parts, even to get the chemical signaling cascade going. Somehow that electrical signal, the electrical signal that comes down axons, has to trigger all the stuff that's happening at the synapse, because neurotransmitters and receptors, all of that was a chemical event. So right away, you realize, even from the very get-go, that these things are totally intertwined. Even from kicking off the whole chemical signaling cascade, we had to somehow do that and I was just telling you mysteriously that the electrical signal triggers the release of neurotransmitter from these vesicles in the axon. And again, I also told you that I can do a whole lecture about how that happens, and I don't think I'm going to do that now. Maybe I can answer questions about that later, about how exactly that happens. But the electrical signal comes down the axon. So now we're going to back up to our starting point, where we have to start talking about this electrical signal. What is it? So electrical signaling. So electrical signaling is happening down this axon. We know that. And what is it? Now remember, it just like we did for chemical signaling, I prefaced the whole issue of why all this stuff evolved in the first place, because the cell imprisoned itself, it protected itself and imprisoned itself with this lipid bilayer, where no water stuff or little particles that are dissolved in water can get across through a lipid bilayer. So if this is the outside of a cell, outside, and all this is inside a cell, this bilayer would go on throughout, it covers the entire surface of the cell. So all of this is lipid bilayer that covers, that literally surrounds the entire tube of the axon, and it's made of these little fat molecules. Remember, they have the fatty layer that does not, that just like it's an oily layer that oils, just like true to form, oils and waters don't mix, they don't dissolve into each other, they're not free to just like run around, and the molecules are not happy to be together. They would prefer to have, the fat molecules would prefer to just essentially interact only with fat molecules, and they exclude the water molecules, and anything dissolved within those water molecules are sort of excluded from that fatty area. So cell membranes are made of a lipid, of a fat bilayer. It's perfect. That's exactly what you would want to do to kind of wall yourself off, to preserve and maintain your insides, keep your home warm and comfortable, and everything happening just like you like it, and maintain control. You can't just have the doors and windows open to the bad elements outside where everything's free to just come in willy-nilly. So plasma membranes facilitated the need for receptors, things that could traverse this lipid bilayer from outside to inside, so that molecules that would bind here can trigger like little conformational changes into the inside of a cell, and then we were setting off biochemical cascades. Electrical signaling also you have to understand the lipid bilayer, because when I get into what the electrical signal is that's coming down here, I'm going to get into that now, but it also involves like something going on with this lipid bilayer. So we're coming to that. So already there's going to be a lot of similarities where the basic structures that we were talking about that determined like a lot of the properties of chemical signaling are also going to be relevant structures and key structures for electrical signaling as well. So, but before I do that, I do want you to understand, before I just say an electrical signal is happening or electricity is flowing through, let's talk about it. So, what is electricity? What is electricity? So, whether you're talking about, so electricity is something that flows or diffuses, if you want to use a more technical term for like, how things flow and like how real molecules flow, you might talk about diffusion, but that's just, diffusion is like random motion of molecules, so they can rock and roll from place A to place B, so they can flow from point A to point B. So something that flows. But what is flowing? Whether it's a copper wire in your house connected to the light switch, or whether it's an axon, what's flowing is charged particles. Charged particles that flow. So if I have a charged particle, and charged particles can come in, and again, so this is interesting. It's like we're going to start talking about like a core thing of like biology, like how a brain works and how it's using electricity. But right away, to understand that, we're kind of coming back to other areas of science like chemistry and physics. So charged particles come in two flavors. There's plus and minus, okay? Just like on your battery or in a magnet, you have a plus pole and a minus pole, and electric fields are a little bit different. That's a little bit, it's different, and yet when you really get deeper, it's magnetism is another manifestation of the same force and field. So physics doesn't treat electricity and magnetism differently. When you really get down to it, those two forces, the electrical forces and magnetic forces, are sort of unified into one electromagnetic force. But in our case, we're just going to talk more about like sort of classic like electric fields. So where you can have positive charged particles, negative charged particles, and you remember even with chemical signaling, when we were talking about phosphorylation, and I showed you this little phosphate area, that this is what gets stuck onto proteins, and I told you that this end of the like these oxygen atoms are sort of like stealing away like negative charges to this end of the molecule. They're like, so that if you're looking at a phosphate group, there's like an electric field, a negatively charged like region of the of the molecule that gets established here. And that's because atoms, if this is like the nucleus of an atom, it has charged particles in it. It has like a bunch of pluses in a nucleus, and then outside flying around, you've got a bunch of negative charges. These negative charges are electrons, and these positive charges that are in the nucleus are protons. And all atoms are made up of just this thing. The only thing that distinguishes a carbon atom from a nitrogen atom, and I actually drew a carbon atom here because there's 6 protons. That would define a carbon atom. But anyway, the only thing that makes a carbon atom different from a nitrogen are just the number of protons in the nucleus. And because protons are positive charge and electrons are negative charge, in a neutral atom, the number of protons equals the number of electrons. So a neutral carbon atom would have 6 protons in the nucleus, 6 electrons flying around, although I think I drew 7 out here, and that's that. So because these atoms this molecule is made of atoms stuck together, and these atoms flying around on their outsides are all the electrons. When atoms of different kinds come together, some are hungrier for these electrons than others, and they grab them, they pull them, they hog them. They pull them closer to their end, and that makes molecules have like a charge distribution. Like all the electrons are congregating around these oxygens, and the oxygens are pulling them sort of out and away from from the phosphorus. Like that's usually how it goes. And again, it's getting more into the details of how this is, and chemistry and polar molecules and electronegativities is a whole chemistry lecture, but it's pretty well worked out. And you can predict when molecules are polar and where the negative charges will be around, and what will be like the plus end of the molecule and what will be the negative ends. But atoms are made of charged particles, and that's key. So electricity is when any charged particle is moving. In your house wires, in a copper wire, it actually is the electrons that are moving through the wire. The electrons are just like flying. Like if you've got a big copper wire here and you turn on a switch, these electrons are literally moving through the wire so that you have an electric current. These charged particles are moving or flowing through a wire, and that is electricity. That's good enough. But like I can have charged people. I can have people on a trampoline where they're like having their hair stick up on end, where they have like a lot of excess electrons on their body. And if people like that have excess electrons on their body with these like tiny negative charges, if these people start running, they would actually make an electrical current. Like you would actually, if you had probably crazy sophisticated equipment, you might even be able to measure it. But like literally any charged particles that are moving, if you took a bunch of phosphates, remember these phosphates are negatively charged. They actually are negatively charged molecules. If I took a bunch of these and just moved them through a tube, I would get an electrical current, but the carrier of that electricity would be phosphates, not electrons. But it doesn't matter. Anything that has an accumulation of charged particles on it, that's rocking and rolling, that's moving, is carrying an electric current. All right. So now. And now let's just come back to the biology here. Remember, an electrical current is flowing down this axon. So what is it? Is it those phosphates that are moving down that tube? But this is nice. It makes it easier. It's like any chemical that is flowing down this thing that's charged is going to be good enough to carry that electrical signal. So you should be asking, oh my gosh, I'm understanding something. What is the electricity? Like, what is the nature of that electrical signal going down the axon? And I will tell you now. So if you take salt, which is made of sodium chloride, and you just take like table salt, so it's a solid, and you put it in water, it dissolves. The sodium chloride particles like end up dissolving out in the water. And when they do that, what happens is they form charged particles. You get inside the water, the sodium and chloride atoms split apart from each other, and you form a positively charged sodium atom and a negatively charged chloride atom. And atoms that are charged are called ions. So charged atoms are called ions. So this is a sodium ion, and the sodium ion is positively charged. This is a chloride ion, and the chloride ion is negatively charged. And when you're drinking salty water or you're in the ocean, it is a giant pool of atoms and it's not just sodium and chloride. There's probably calcium and potassium and all sorts of other things mixed into into an ocean and in your blood. There's all sorts of atoms that are dissolved in there, and most of those actually exist as ions, as charged particles. So, and again, I can sort of describe, like, when the sodium and chloride are together in the salt, when they come apart, I can sort of tell you what happens. You can guess what happens. Since the chloride ion is negatively charged, when these guys break apart from each other, the chloride ion steals an electron from the sodium. So the chloride breaks away with one extra electron on it. So it's negatively charged. And because the sodium had it stolen, but it still has its positively charged protons in its nucleus, it's got an excess of positive charge. So your sodium is left with an excess plus. Your chloride, because it stole an electron, it hijacked an electron, it's got that, and it's a Cl minus. A chloride ion has a minus charge. So for the most part, the ions that are flowing through the axon is sodium ions. It's mostly sodium ions. So you get a positively charged flow of sodium ions that's flowing down this axon, and that is an electrical signal. Your blood and your body and your cells, every piece and part of your cells, whether it's inside of a cell or outside, has tons of these ions. So you see, if you're and ions because they have charges, just like we talked about with the phosphate, when you can do chemistry, when you can make like little structures that have like force fields, that have like charged areas or charges on them, you can do stuff with them because you can do physics now. There's an electric field that you can use, that the cells can use to their advantage, to push and pull things and to change protein conformations and all sorts of things. We've seen this before, this fundamental aspect of chemistry and physics that is just being hijacked. And it really makes you realize that living things are really just chemistry and physics machines. They're doing basic chemistry and basic physics, and they're hijacking nothing like that's new in the universe. They're hijacking tried and true physics principles to do their function. And that's exactly how I at least view living organisms. It's a very reductionistic approach and it's very threatening to a lot of people, but whatever, I'm not apologetic about it. That's how I view it. So the electrical signal has to be a moving charge particles, moving charges. But now you're getting very familiar with a lot of molecules and ions exist that way in biochemical systems, inside cells and in the blood. And where I'm going to start going next is I've told you pretty much that it's sodium ions for the most part. We're going to get more into detail, but it's a sodium current. That is kind of the primary thing. That's that's sort of a lie, and I'll have to kind of really flesh that out a little bit more to kind of so that that statement doesn't get too misleading. But for the most part, I think I'm happy enough to say that it's a sodium current that moves down this axon that will end up triggering the other things. But I just want you to be familiar now with a little bit of the chemistry and physics and charges and understanding that electrons and protons are the particles that make up atoms. There's neutrons, but they don't have a charge. They're also in the nucleus. The electrons are negatively charged. The protons are positively charged. Because these things make up atoms, and atoms make up molecules. So it's a beautiful system. You've got because you've got charged things, you've got all these things that can work together or that can induce conformational change. If I have a protein that has a surface that looks like this, and let's say it's a positively charged surface because the electrons from these atoms have been pulled away over here. And now I've got another protein that is shaped like this, but it's negatively charged. You can see how protein machines can come together and stick together and interact. This is just like plus minus, like charges interacting and shapes that are folding into each other. So this defines the nature of how molecules can interact with each other. It's a huge idea, and it's nothing more than just chemistry built up on simple building blocks of those big macromolecules that make up your cells. So that's all I want to do, is just get you familiar with the charges, and then I'm going to start to talk about now. So let's say so these charges were flying through, but what's pushing them? Now there's still a lot of mystery here. What is pushing them through? How can you create a pushing machine for, let's say, sodium ions? And also I'm going to start moving into an area where, even though there's a chemical signal, I told you about chemical signals get kind of sent down through signals through the synapse. It's also the case that there's electrical signals. You can see, for the first time, I'm going to pay attention. You see these pluses here in this diagram? This is our first hint that somehow in the chemical transmission at the synapse, it's not just the biochemical cascade, there's also an electrical cascade that makes its way through. So but now you've got a toolkit to understand how that happens. So there's an electrical signal coming down, and we're going to learn in the next videos how now that electrical signal carries through down to this cell, and what that looks like, because now we're going to have to transmit an electrical signal through a membrane, but we still have the problem because a sodium ion trying to get through on its own is not going to be able to. It can't get through the lipid bilayer, so you should know. Of course, you better be thinking ahead and say, okay, there's going to have to be some other kind of receptors or proteins that are going to trigger the electrical flow to continue on into cell number 2. You will see that that's the case. So again, I'm just slowly building on what you're learning, on all these complicated details. And you are learning a lot, and I'm trying not to rush it. This is probably painfully slow and boring, but if you're staying with it, hopefully you're learning. I'll do more next time.